Hi, I'm Jens xenograft from the TU Dortmund University.
Hello everyone, I'm Sven Rahmann from the University of Duisburg-Essen and I'll be giving the talk.
The official title is Fast, Lightweight, Accurate Xenograft Sorting. To explain a little bit the background, we are talking about patient-derived xenografts. The idea is that we take a human tumor or a tumor cell line and engraft it into mice. So we have mice with human tumors. And then we can test several substances for therapy, or we can study tumor evolution under different conditions. So after a while we would take a biopsy and we sequence it. Typically we then have a mixture of the human tumor reads and some of the surrounding mouse tissue reads. The typical first task in this case is to clearly separate the human tumor reads from the host mouse reads. This is not a trivial problem because the human and mouse genome are very similar. This can even lead to so-called human-aligned mouse alleles or HAMAs, which were described recently. The authors here showed that reads that actually belong to the mouse genome can be falsely aligned to the human genome and then give rise to false variant calls in the human genome. And this is of course a problem if you study mutations induced by the tumor. The xenograft sorting problem then can be stated as follows. We are given a sequenced xenograft sample. So we have reads from 2 different species. We would like to sort the reads into 5 different categories according to the species of origin. The host genome, so typically mouse, the graft genome, typically human, reads that could originate from both. So if the sequence is identical, we cannot really tell. Could also originate from neither species, so this could be technical artifacts or contamination. The 5th category is actually ambiguous reads, where we get mixed signals and cannot really make a good decision. And there are several variations of this sorting problem. Sometimes we might only want to count how many reads are in each category. And of course there are already many tools that solve this problem. There are 2 different kinds of tools. Some tools that require to run a mapper and aligner first, and these will work on aligned BAM files. The other category takes the raw FASTQ data as input and does not require alignments. A good way to do that is to work on KMeans. KMeans are short substrings of length k, so we partition each read into its KMeans. Here is an example for k equals 5. We have a large table that associates to each KMean the species it originates from. If we have processed a read, we can make a statement about the read, for example, by a simple majority vote. While this seems conceptually simple, the devil is again in the details, because we have 3 conflicting goals. We want to be fast, we want to be lightweight, which means to have a small memory footprint, and of course we want to be accurate, so we want correct classifications. And for each of these 3 goals there is a limiting factor. Obviously we can't be as fast as we want because we have to look up the associated data for each KMean, and the random memory accesses are slowing us down. So a random memory access is very slow compared to a simple arithmetic operation because it can take up to 300 or 400 processor cycles. So to look up KMeans we use a very specially engineered form of hashing which we call 3-way bucketed cuckoo hashing, which we'll explain in a moment. The KMean index also can't be as small as we would like it to be because we actually have to store a large number of KMeans, which is around 4.5 billion for each human and mouse KMean that is in the genome or transcriptome. The only way we can deal with it is to use an almost full hash table, which is one of the properties of this engineered Cuckoo hash table. And we also use a technique called Quotienting to reduce the number of bits we actually have to store per KMean. If you want to be accurate, the limiting factor is that human and mouse have KMeans that are similar to each other, and we use an idea from genome to mark them as weak. And also we have a sufficiently complex multi-level decision rule. Cuckoo hashing basically means that you have a fixed number of hash functions, usually 2, but in our case 3, and you're guaranteed to find a key, in this case a KMare X, at one of the locations pointed to by the 3 hash functions. So if X is in the hash table at all, it is either at location F1, F2 or F3 of X. In our case, each location in the hash table does not only store a single key value pair, but up to 4. So each location is a bucket that allows several elements. And one of the key ideas is that each of these buckets fits into a cache line. So once we do a lookup of one element in a bucket, essentially looking up the other elements here comes for free. So while we have 12 possible locations for each key value pair, we only need at most 3 cache misses, and on average it's much less. It's only around 1.2. So of course, if the lookup is quick, there has to be a disadvantage, and the disadvantage is that insertion of elements can get complicated. So obviously we first try the first bucket, then the second bucket, then the third bucket, and whenever there's a free space somewhere, we can just insert the element at the first free location. But what if all of the possible 12 locations are already full? Well, we pick a random element that is already there, like this one here, and we remove it from the table to insert X, and then we deal with the element that we have just taken out and insert it at one of the other 11 alternative locations for this one, some of which might actually be free. If not, we repeat the process. So we construct basically a random walk through the table, examining different elements till we find a free spot. And in order not to take too much time, we limit the number of steps of this random walk to a constant number. And if that doesn't work in this number of steps, we fail and say the hash table is too full. Now, interestingly enough, this allows for quite a high load factor. Of course, the random walk is not an optimal insertion strategy. We can, in fact, solve the combinatorial optimization problem that asks us to place each element optimally in the hash table. And by optimally, I mean to try to place as many elements as possible at their first bucket. So the bucket that's pointed to by the first hash function, and that will therefore be examined first. So that incurs only a single cache miss. Now, we showed previously this year at Alinex that this optimization problem can in fact be solved on a genome scale. We here show the average lookup cost in terms of the number of average cache misses for different load factors of the hash table, for different bucket sizes from 3 to 8. In practice, for the xenograft application, we decided to use a load factor around 88% and a bucket size of 4, which gives us around 1.1 cache misses per lookup on average for the optimal placement and around 1.2 for the random walk insertions. So the gap is not too big here. So this is a good compromise between a fast lookup with a small number of cache misses per KMare and a high fill rate for the hash table. We store a little bit more information than just the K-mares and the associated species. We also mark K-mares that have a Hamming distance 1 neighbor in the other species as weak, because in this case a single nucleotide variation suffices to switch the species. So we don't have 3 different values for each K-mare, but 5. So we have host K-mares, graft K-mares, and K-mares that appear in both. We also have weak host and weak graft. Of course, this requires finding all the Hamming distance 1 neighbors of each K-mare in our table. And in the paper, we describe a fast method to mark the K-mares as weak. Now let's talk about saving space with a technique called quotienting. So our keys are canonical codes of 25 mares, which need 50 bits, and we have 5 distinct values, so we need 3 bits. As we have around 4.5 billion K-mares in all of the reference genomes with alternative alleles and transcripts for human and mouse, and need 53 bits per entry, at our load factor we need almost 34 gigabytes for the hash table, which is too much still for a typical desktop PC. But there is this technique called quotienting, which reduces the number of bits that we need to store for the keys from 50 to around 20 bits plus 2 bits to remember which hash function choice we used. And again, the value bits, so we need only 25 bits per entry. And then suddenly the hash table fits into a slightly less than 16 gigabytes of RAM. Remember that DNA KMeans can be base 4 encoded and represented as integers between 0 and 4 to the power of k. To define a hash function, we proceed in 2 steps. So it's important that the first steps is just a bijective map from this set of integers up to 4k to itself. So we use a simple hash function that first rotates the bits of the key, then XORs in some arbitrary number and then multiplies by some arbitrary odd number. So the hash function is parameterized by 2 integers and then takes the result again mod 4k. And because a is odd, it has a multiplicative inverse, and so we can show that it is bijective. Only in the second step we take the resulting value and take it modulo the number of buckets p to obtain the bucket address, and at the same time we take the result of the integer division by p, so the quotient. This has the effect that we can reconstruct the k-mer x from the bucket number f of x, so its address, and the resulting quotient. Now because we store x at address f of x, we have implicitly encoded a part of x in the bucket number, and we only need to store q of x, the quotient, explicitly. And this is where we save a number of bits. Now very quickly about build time and memory. We need less than 1 hour to insert all the k-mers into the hash table, but we need more time to mark the weak k-mers because we need to compute the Hamming distance 1 neighborhood for each k-mer. Overall, the hash table is finished, including the marking, in less than 4 hours of wall clock time, needing less than 16 gigabytes of RAM, with a slightly higher peak during the marking phase. The directly comparable tool Xenome needs much more CPU work, but is better parallelized during insertion, still needing 3 times as much time as we do, and needing much more time for the marking phase, also needing more memory finally and in the peak. Moving on from building the index to using the index for read classification, we partition each read into its valid k-mers and then just count how many k-mers belong into each category. We have a complex decision rule. So for example, if we have no graphed k-mer at all, and then a small number of host or weak host k-mers, then we say it's host. Well, you can see that the rules are quite complex and there are many details in the paper, so I will not try to explain the details here. We implemented a heuristic called quick mode. Here we only examine the third and the third last k-mer in a read or in a read pair and look up their classes, and if they agree, we directly classify the read accordingly, and if they don't agree, then we use the decision rule. Let's discuss some results. We compare the alignment-based tool xenofilter against the alignment-free hashing-based tool Xenome and our tool xengsort, which implements all the techniques I mentioned so far. Of course, we should classify pure human datasets and mouse datasets correctly. So here we take a well-known genome in a bottle human mate pair dataset, and we can see that our tool xengsort classifies almost everything as graphed, so this is human. The same holds true if we use the quick mode, and Xenome classifies a little less as graphed, but slightly more as ambiguous. Now all of the tools classify some of the sequences as neither. This is also correct because there is some phi x that has been added to the sequencing library, so most of these reads are actually phi x. Similar results hold for pure mouse datasets. It's also interesting to ask what happens if you input nonsense, so if the tools are actually robust against noise. So we use a chicken dataset. Here we also compare the xenofilter tool, which wasn't able to deal with the size of the human dataset. Now xenofilter only extracts graphed reads, so it does not classify the remaining reads. So it is absolutely correct that we see an empty bar here for graft. Our tool Xengsort thinks there are very few human reads. It does classify most of the reads as neither, which is correct. Xenome distributes around 10% across wrong categories and only classifies around 90% as neither. Finally, let's look at a real application data set. We obtained 174 RNA-seq patient-derived xenograft samples from an internal collaboration partner, Jens Sieweke. Again, xenofilter only extracts the human reads and finds around 70%. Our Xengsort and Xenome find slightly more. Again, we see the effect that Xenome classifies more reads as ambiguous because it has stricter thresholds. We also see around 25% of mouse reads with each tool. In the end, it appears that xenofilter is missing some of the true human reads. What's more interesting from an algorithm engineering standpoint of view is the running times here in total CPU minutes. So this is the sum over all 174 data sets. Now, if you look at the xenofilter tool, which only compares to BAM files, and compare it to Xengsort, which does the full alignment-free analysis, you find that even scanning the BAM files is slower than doing the full sorting from the raw data. If you add the time required to do the alignments and to sort the BAM files, of course, using an alignment-based approach takes forever. Xengsort should be compared to the other alignment-free tool, which is Xenome, but here you see that it's over 4 times slower on these data sets. We also used the count operation of Xengsort, so remember it does the same thing except it doesn't produce the output FASTQ files. So we see that's not the majority of the time spent for output. We looked at the variant where we optimized the hash table. The effect is that more of the KMeans can be found at their first hash function, so we need fewer memory lookups. And overall, this is a measurable effect. We save about 2000 CPU minutes in comparison to the random walk produced hash table. Then we asked what happens if we need to save some more memory. So if we moved from a fill rate of 88% to a fill rate of 99%, of course, then we can place less of the elements at their first hash function. So we have a more even distribution of elements over their 3 possible hash functions. So we are a little bit slower. Then we can, of course, run the placement optimization also on this 99% loaded hash table, and then we find, well, of course it's a bit slower than the optimized 88% load hash table, but not that much, and it's still faster than the standard 88% load random walk build hash table. And then we looked at the quick mode, which is of course much faster, but maybe not so much faster as you would expect, because it still needs to do the I/O and compute KMeans and some hash functions. So it only takes about half the time of the standard approach, but not much less. We have engineered an alignment-free approach for the xenograft sorting problem. It's lightweight on CPU resources because it uses a 3-way bucketed Cuckoo hash table. Our implementation of this outperforms an existing tool which does a very similar thing, but we need only 1/6 of the CPU work and 1/3 of the wall clock time over different data sets. We also find that typically it takes the same order of magnitude of time just to scan the BAM files, so there is really no advantage in using an alignment-based approach. Now our table can fit into 16 gigabytes of RAM, so the approach can be used on a standard desktop computer. We summarize again the algorithm engineering aspects, so we can really recommend the 3-way bucketed Cuckoo hashing with a bucket size of 4 to store many KMeans in a compact and fast accessible way. So we get the table small by using the quotienting technique, where we store part of the key implicitly in the bucket number. We can use smaller or larger buckets. If we use smaller buckets, we also have smaller quotients, so we need less memory per key, but of course it also limits our possibility to distribute keys, so we get a smaller maximum load and fewer keys at the first hash choice, so lookup becomes slower. If you would like to try out Xengsort, here is a GitLab link. Thank you.